I'd like to start by acknowledging the traditional owners of the land on which we meet, the Wurundjeri. I acknowledge their history, their elders past, present and future, and I think it's appropriate in the light of our having very recently celebrated Anzac Day that I acknowledge the role that Aboriginals have played fighting alongside their fellow Australians in conflicts past and present. Before we get on to the main bit, am I clear or am I very muddy? Okay. Before we get on to the main business of today, just a few words about MS Plus, which, as many of you would know, and some of you probably don't, has been in the process of selling the nerve centre at Blackburn. And they've just recently announced that they have successfully completed that sale, and they've taken up what I assume is a lease in Box Hill. Sorry, in Hawthorne. We're in Box Hill. In Hawthorne, close to Camberwell Junction. John Blawinski, the CEO, said that the sale marks a significant milestone in MS Plus's long-term strategy to ensure every asset is working as hard as possible. And I would have to acknowledge that the nerve centre has been much larger than they needed recently, and it was an enormous site with a large car park attached. So I'm not at all surprised that they've realised the capital value of that. The new premises, according to John, have been selected with clients in mind. It is accessible. It has drive-in, drive-out access, dedicated disability parking, and good public transport links. I'm not familiar with the building myself, but hopefully it will live up to what John describes. Now talking about today. Today's presentations will all be video recorded and posted on the website, so that you will be able to re-look at them if you wish to, or recommend or recommend to anyone else that they look at it. And in fact, at least myself, considering the possibility that this might be the last conference we hold in this form, because social media is making it so easy for people to view things online. And in fact, we're increasingly holding our committee meetings on Zoom, and so I think it's quite possible that we might move to a situation where we do a series of talks over the year and put them all online. We have 3 presentations today, and to some extent they follow a theme, and that is managing or reducing the impact of MS. The first presentation by Sarabeth Stone, Dr. Sarabeth Stone, will talk about the search for something which might reduce or even repair the damage done by the disease to the myelin sheath around the nerves of the central nervous system. The second paper will discuss the benefits to be gained from socialisation and lifestyle factors, mixing with people, exercising, having the right diet and such like. We know that optimism and socialisation and diet can have a significant impact on MS. But I have to say, I think it's an under-researched area. and I wish there was much more information available about it. The third paper will talk about a completely different topic, and that is travel with a disability. Again, I'm quite sure that travel is good for MS to the extent that it is possible. The mental stimulation that comes with travel and the socialisation which comes with travel must be beneficial. And again, it's an area which I suspect is under-researched. Our first presentation this morning, Dr. Sarabeth Stone. She's postdoctoral research officer at the Florey Institute of Neuroscience here in Melbourne. A senior research officer within the multiple sclerosis community. Sarabeth says she was inspired to get involved in MS research and became fascinated with the immune system in neuroscience, but more particularly because her mother has MS, and so she has seen firsthand the way MS affects a person's life. Her current research focus is something called microRNAs, a small molecule produced within our cells that controls cell function by regulating gene expression. And she says it's dysregulated in MS. The aim of her research is to identify any that might promote remyelination. And let's face it, MS is a disease in which the myelin sheaths surrounding the nerves of the human central nervous system get damaged. And finding something which might prevent that, or something which might repair the damage, is perhaps the holy grail of MS research. So I'm looking forward greatly to hearing from Sarabeth. Over to you, Sarah.
Thank you, Nigel. Can you all hear me? Wonderful. So I would also like to begin by... the microphone's not working? I think I'm mic'd up. These ones? Okay. How's that? Better? All right. So I would also like to begin by acknowledging the traditional owners on the land in which we meet today, the Wurundjeri people of the Kulin Nation, and acknowledge their elders past, present, and emerging. So as Nigel said, I'd like to start with a little bit about myself. So my name is Sarabeth Stone, and I'm originally from Wellington, New Zealand, and I have been an MS researcher for more than 15 years now. My journey in MS research started in 1996. Might be better with this? All right. Is it on? Yes. All right. Better? Wonderful. So my journey in MS research started in 1996 when I was 9 and my mother was diagnosed with MS. And in preparing this talk, I realized that it's been 30 years now since she was first diagnosed. And it's been a long journey for our family, but she's doing relatively well, all things considered. At the time of her diagnosis, treatment options were very limited, and it would be another 4 years before disease-modifying therapies would be available in New Zealand. And because of this, I developed an interest in multiple sclerosis. I pursued immunology and neuroscience throughout my undergraduate and eventually my PhD, where I researched ways to control brain inflammation. In pursuit of this, my studies took me halfway around the world to America and finally back here in Melbourne, where I am a researcher at the Florey Institute. Slow down? I'm sorry. I'm a little nervous. I'm a researcher now at the Florey Institute. So we are, We are the largest brain institute in the Southern Hemisphere, and our mission is to improve the lives of people through brain research. I have had the privilege over my career to witness many new breakthroughs in the field of MS, not just in understanding the disease, but also in patient care, advocacy, visibility, and community. And of course, developing new treatments. It's no understatement to say that the treatments that we've developed for MS have changed lives and conversations about MS. However, we still face many unknowns. As a brand new PhD student, I had been watching my mother live with MS for more than a decade. She responded well to treatment, her relapses were well controlled, but she was still progressing. And no treatments could reverse her symptoms, and this is what has driven my research. My question since then has always been, why is there progression, even when relapses are well controlled, and how do we repair the damage in a way that produces real functional change for people with MS? As it turns out, there are two sides to MS, one that can be addressed well with our current treatments and one that cannot. I'm here today to talk to you about the other side of MS, beyond the relapses, and how we hope to be able to harness brain cells themselves to protect and repair the brain. No? Yes? Please? Sorry? Point? I wonder if I can do it here. Nope, that's the volume. Point that way? Oh, good? Okay. Do I just need to tell you when to click, or will I be able to? Oh no, I can do it. Okay. It's working. Yeah, thank you. So if we were to give MS a technical description, it would read something like this. MS is a chronic, autoimmune, demyelinating, and neurodegenerative disease of the central nervous system, which is a lot of words all in a row. And that's because in some ways MS is a lot of different things. And today we are going to be discussing some of that diversity. Briefly, what happens in MS is that there is damage in what we call the central nervous system. That's our brain and our spinal cord, the central control hubs of our body. This damage to our brain cells interrupts the ability of our brain to communicate with the rest of our body, leading to a wide variety of symptoms. Not everyone experiences every symptom, and these percentages show the proportion of people who report having these symptoms in MS. Some of them are very, very common, like walking disabilities, pain, numbness, and tingling. Some are much less common, but as you can see by one of the least common being seizures, their lack of commonality does not make them any less serious. This image doesn't just show the wide variety of the disease, but it also indicates just how diverse the symptoms are. Sorry, I lost my train of thought. Different parts of our brain control different parts of our body, and a large reason for this wide variety of symptoms comes down to where these lesions appear within our brains. MS occurs worldwide, but has a global distribution where the incidence is higher in countries further from the equator. The darker the red, the higher the incidence. And you can see from the dark red in Europe and America, and also in Australia, that countries that are further from the equator have higher prevalences of MS. It is believed that a strong contributor to this pattern is sunlight exposure and thereby vitamin D levels. However, it's likely that other factors, including genetics, are also playing a role in this pattern. In the last report that was conducted by MS Australia and the Menzies Institute, it was revealed that in 2024 there were almost 38,000 people living with MS in Australia. And this prevalence has been steadily increasing since 2021. If our current trends continue the way they have been, The 2010 numbers will have doubled before we reach 2030, which is quite a dramatic statistic. But I do want to take a little aside here, though, and talk about one thing that might be increasing the prevalence in the next coming years. There is something called the McDonald criteria, which is what neurologists use to diagnose MS. And it was updated in 2024. One of the major changes that was made in the McDonald criteria now allows people with MS to be diagnosed earlier, which may have an increase in the prevalence in coming years, raising it. However, I do want to say that this is actually a really good thing. We know that one of the best things that we can do for people with MS is to get them on high efficacy treatments as soon as possible. And the changes to the McDonald criteria are going to be helping that happen, which is only going to be beneficial for people who are newly diagnosed with MS. So onset of MS is usually between the ages of 20 and 40, and that makes it one of the most common causes of non-traumatic disability in younger adults. It affects approximately 3 times as many women as it does men. However, these statistics that you commonly see are mainly for relapsing forms of MS. People with progressive MS tend to be diagnosed slightly later in life, with the peak being closer to 40 years old, and it affects an equal number of women as it does men. The cause of MS is complex because it's not just caused by one thing. We know that there is some genetic susceptibility and that there are certain environmental factors that affect MS risk, like low sunlight, obesity, and smoking. Epstein-Barr virus has recently been determined to be a key factor for developing MS. Most people will develop a Epstein-Barr infection at some point in their lives, but only a few of those people will go on to develop MS. But if you don't get Epstein-Barr, you won't get MS. So it seems that it is what we call necessary but not sufficient for an MS diagnosis. So multiple sclerosis has multiple clinical courses, and you may well have seen graphs like these before, which show an overview of the symptoms that the symptom severity that people with MS experience, where disability is changing over time. The majority of people with MS, approximately 85 to 90%, are diagnosed with a relapsing form of MS where they experience periods of elevated symptoms followed by periods where those symptoms resolve. Some of these people will then go on to experience progression without relapse, which is termed secondary progressive MS, and finally there is a primary progressive MS where progression of symptoms occurs without relapse from the onset. What I want to draw your attention to in this slide is these parts here in relapsing MS, where progression is occurring without relapse. It turns out that people with relapsing MS can experience progression that is associated with relapse, where their level of disability elevation when they enter remission compared to what it was before the relapse is elevated. And we call this relapse-associated worsening, or RAW. However, people with relapsing MS can also experience a disability progression between relapses, even when their relapses are well controlled by therapeutics. And we call this progression independent of relapse activity, or PIRA, and it's similar to what is happening in progressive MS. In our current age of high efficacy treatments to target relapses, progression independent of relapse activity is now more common than disability due to relapse. Current treatments do not have a large effect on this type of progression. My research is focused on both preventing this progressive phase, which is experienced by people with both progressive and relapsing MS, and to find ways to reverse this damage. The reason that there are these 2 distinct phases is because there are 2 very different things that are happening in the brain. And to explain this, I'm going to go a little deeper into what is happening in the brains of people with MS. So within our nervous system, our brain cells, called neurons, transmit information to other cells within our brains, down our spinal cord, through our peripheral nerves, and eventually to our muscles, turning our thoughts into actions. To do this, Neurons extend long fiber-like processes called axons through which they send electrical signals. Some of these axons are really short, they're talking to the cells next door, they're just a few millimeters, but other ones extend the entire length of your spinal cord and can be up to 1 meter long. And so to get the message from your brain through your spinal cord quickly, your nerves have to be firing fast. And the way they do that is through myelin. Myelin insulates the nerve and provides these little spaces where there's gaps in the myelin. And that allows the electrical signal to jump fast down the axon, delivering the messages quickly. When a neuron doesn't have these fast axons, it's slow. It takes a lot longer for the message to get through. It's harder for our brain cells to do it, and it takes them, sorry, I'm talking fast again, and it takes them a lot more energy to be able to get these thoughts through. In people with relapsing MS, they have what's known as an autoimmune inflammation against the myelin sheath. This is where your body's own immune cells come in from outside of your blood and incorrectly see the myelin in the brain as a threat. They view it as something like a bacteria or a virus, something that's not supposed to be in your body, and they attack it. This causes damage to the myelin and eventual loss of the myelin. This means that those electrical signals can no longer travel fast. It takes more energy for our brains to do the things that it had been doing before. And it also leaves the axons and the neurons vulnerable to damage. It is possible for myelin to repair itself, which protects the neurons and restores their function. However, this process becomes less and less efficient the longer a person has MS, but it also becomes less and less efficient as we're aging. It happens with everyone, that this process becomes worse, basically. We have developed, and if the myelin cannot be repaired, it typically leads to degeneration of the neuron. We have developed many treatments for MS which are targeted towards this autoimmune response. And finding ways to promote this myelination is one of the most promising strategies to protect the brain. This plot is showing some of the major breakthroughs in MS treatment. Prior to the early 90s, there were no specific treatments for MS. When an inflammatory relapse, steroids could be applied to dampen the autoimmune response, but that was all. In 1993, the first specific treatment was approved by the FDA in the United States, which was followed a few years later by Copaxone. In 2024, Tysabri became our first high-efficacy treatment. In 2027, Ocrevus became the first treatment approved for primary progressive, and in 2019, Mayzent became the first approved treatment for secondary progressive MS. There are now more than 20 approved drugs by the FDA in America, although I believe only 14 of them are available here in Australia. We've clearly come a long way in the last few decades. And while a lot of these treatments are working really well at doing the thing that they are designed to do, they mainly target the autoimmune response in one way or another. Treatments like interferon gamma and Copaxone tweak the immune system ever so slightly to make it less damaging, so that when the immune cells get into the brain, they're doing less damage. Other treatments like Tysabri and Gilenya prevent immune cells entering the brain at all, so that they can present damage. And while these treatments have been revolutionary, they don't work for everyone, and most of them don't have a significant effect in primary or secondary progressive MS. And this is because of something that is going on within the brain, separate from the autoimmune response, and we call this smoldering inflammation. And that is what's driving neurodegeneration. In smoldering inflammation, it's not the cells from outside of the blood that are causing damage, but cells that are already in the brain are causing damage. These cells make an environment that not only promotes damage, but prevents repair from happening. making neurons more susceptible to generation. We call it smoldering inflammation because it is, by comparison, low level and slow. It doesn't cause the same relatively dramatic flares and symptoms that you see in relapsing MS. And because of this, and because we have not worked out how to stop it, it continues to slowly cause damage. We often view smoldering inflammation as the bottom of the iceberg in MS. It's much less obvious to see, but we can't treat it. And finding ways to repair myelin is going to be one of the best ways to address smoldering inflammation. And I am interested in looking at this by looking at a type of cell called a microglia. So microglia are the cell type that is causing all of this trouble in smoldering MS. They are the only brain cell that lives in the brain full time, all the time. And just while I have talked about them being damaging in smoldering MS, that's not actually what their normal function is. Microglia are caretaker cells. They scan the brain for infections and damage. They remove waste from the brain. And in normal circumstances, one of their jobs is to repair the brain. The problem is that in MS, they have been chronically activated, and these microglia become neurotoxic and cause damage. But they have the capacity to protect and are essential to the repair of the brain. My research focuses on understanding the microglia understanding the ways to manipulate microglial function to promote repair of the brain instead of causing damage. In this way, we are hijacking the brain's own repair system to unlock and enhance the function of these cells. Microglia promote myelin repair in several different ways. They're known as a type of cell that we call a phagocyte, and that means it's an eating cell. So one of the main things they do is they remove damaged myelin. You need to have myelin removed, damaged myelin removed in order to lay down new myelin, the same way that you wouldn't paint over flaky paint without removing it first. In order to lay down healthy myelin, it has to be removed. And one of their jobs is to remove this damage so that healthy myelin can be restored. The second thing they do is that they control inflammation. When they are behaving themselves properly, microglia tell those immune cells that are invading into the brain that they need to calm down, that this isn't something that they need to be worried about, and that can help reduce damage. The third thing that they do is that they directly promote the growth of myelin. Myelin is produced by a cell called an oligodendrocyte, and microglia produce growth factors that help them proliferate, grow, and develop, and produce mature myelin. I am interested in a small molecule called a microRNA that we know is critical to microglial function, and finding ways to utilize this to promote repair through microglia. So microRNA are these tiny, tiny control switches that are produced in every cell in our body, and they control the function of every cell, including microglia. They function by disrupting gene expression. So what would normally happen is that a DNA will make an RNA, which turns into a protein, and then that protein has a function in the body. And often those functions in disease can be pathogenic. Multiple sclerosis, microRNAs interrupt this process by binding to the microRNA and stopping it from being made into a protein and destroying the microRNA instead. In this way, they are controlling the way that the cell is functioning by preventing the expression of harmful proteins or by promoting the expression of helpful proteins. We know from studies of people with MS that these microRNAs have become dysregulated, and some that are associated with inflammation have much higher levels than they should have in the brain. However, we believe that we can harness this natural pathway to promote neuroprotective properties of microglia. MicroRNA control so many aspects of microglial function, including the way they are activated, their inflammation, and the clearance of debris. However, there are now over 1,000 microRNAs that have been identified, and we don't know for sure at this point exactly which of the microRNA are most important. And that is what I have been researching, with my aim to identify and characterize the microglial microRNA that promote myelin repair. So to do this, we engaged in collaborations to understand the core microRNA signature of microglia and how it changes across age, disease, and species. In collaboration with clinicians from the Royal Melbourne Hospital and the Royal Children's Hospital, we obtained tissue from surgical resections, which allowed us to isolate human microglia and generate the first comprehensive study of the human microRNA in microglia. We also generated a complementary data set in mouse development and in a mouse model of myelin damage and repair. By comparing these data sets, we have been able to identify microRNA that are key to promoting repair that are also conserved across species, allowing us to ensure they will have translational relevance to humans. Out of more than 1,000 microRNA that exist, we identified those that are enriched in humans, in mice, and in myelin damage and repair. And from these, we identified 13 microRNA that sit at the crossroads of human relevance to disease, and clinical translation. And we're really excited about the therapeutic potential of these candidates. And I'm just going to be telling you a little bit about one of these, which we call microRNA A. MicroRNA A is one of the most highly expressed microRNA in both human microglia and mouse microglia. And you can see their expression of the microRNA is in the red, while the expression in all other central nervous system cells is in the black. And this microRNA was also had reduced expression during demyelination, suggesting that it may have relevance to disease processes. We have been using cell culture assays to understand how this microRNA is working in microglia. And the way we do this is using something called a transfection. So what we do is we take a synthetic microRNA and we introduce it into the microglia where it can act as a natural microRNA, essentially increasing the level of this microRNA within the cell. We've been using a battery of tests to understand how this microRNA affects the ability of microglia to clear debris, their ability to produce inflammation and growth factors, and their ability to promote myelination. To understand how they clear debris, we've been using a fluorescence assay. We culture our microglia and increase the level of our specific microRNA, and then we culture them with a special myelin that has been labeled with a fluorescent dye. This fluorescent dye, when it's taken up by the cell, will glow a bright, bright red, and then we're able to measure how much the cell is glowing to know how much myelin is taken up. We found that microRNA A alters myelin debris clearance by significantly reducing the amount of myelin debris that is taken up in mouse cells, and we also repeated this in human microglia that we had derived from stem cells, showing that this microRNA has conserved function across species, which is essential for translation into clinic. These results suggest that potentially increasing the amount of, potentially reducing the function of microRNA A may be able to enhance myelin clearance, and we're in the process of understanding that now. A second test that I'm going to talk about is how we look at myelin production in a cell culture system. In our lab, we grow neurons in a dish, and we culture them with the cells that produce myelin, the oligodendrocytes. When the microglia are added to these cultures, they support the oligodendrocytes' ability to produce myelin. And we're able to see how microRNA A affects this function. And what we found was that when our microglia had higher levels in the microglia, the oligodendrocytes produced about a third more myelin than they do when they're cultured with control myelin, suggesting that this microRNA may be critical to promoting myelin repair. So to conclude, progression independent of relapse is a primary cause of disability in all forms of MS, and the ability of current treatments to address this type of progression is limited. Microglia, the brain's immune cells, normally promote repair, but in MS, microglia become chronically activated, causing damage. By targeting microglial microRNA, we can turn them back into protector cells. We hope to turn this into a functional therapy to prevent disability and promote brain repair. Before I finish, I would like to thank the other MS researchers from the Florey. MS is complex, but we are trying to come at it from every angle. We want to know what causes it, what triggers progression, and how we can not only slow it down, but reverse it. Science is no longer one guy in a lab with a microscope. It's strengthened by communication and collaboration, and I have the privilege of working with incredible researchers with diverse expertise and perspectives who constantly strive to be better researchers, and they have made me better researchers just by knowing them. I would like to thank the members of our group, both past and present, including Alex Walsh, who worked a lot on this project. Our wonderful collaborators, and also the organizations who funded this research, without whom it would not have been possible. And finally, I would like to thank you all for allowing me to be part of today. One of the most important lessons that I've learned is that science cannot exist in a vacuum. It's better when it's shaped by the people that it's for. Connecting with the MS community, hearing your stories, your frustrations, your hopes, it keeps us grounded in reality. It's a kind of insight that doesn't come from a textbook, and reminds me that it's not all about the molecules and the mechanisms. It's about lives. It's about your lives. And when I listen to what matters to you, I'm better able to ask the right questions and focus my efforts on solutions that will make a real difference, so that I can pursue strategies that are relevant to you. So I would really like to thank you for having me today. And if you have questions, happy to take them.
Questions? I've got a couple of questions we might start with. One is your work looking at microDNA.
Mm-hmm.
Is that doing anything about finding what causes the damage to the myelin in the first place? I don't know whether people could hear my question. What I'm saying is the work that Sarabeth is doing telling us anything about what causes the damage to the myelin in the first place? It's one thing to look for repair, but I'd like to know why is it happening?
I'm not directly working on that, but we do know that the microRNAs are quite dysregulated, and that some of these are definitely driving damaging processes in the brain. We do have some of the other people at the Florey working on just trying to understand the genetic causes of the disease and how those might be changing the functions in the brain, and whether or not those are the key to driving repair, the key to driving that initial damage.
And your work is looking at that smouldering.
Yes, smoldering inflammation. Yes. Damage.
How does that relate to the damage which occurs during a relapse?
So it's sort of separate to the damage that is causing the relapse. So those processes can be going on at the same time. The relapse is caused by the cells coming from the periphery into the brain where they're causing damage, but at the same time there's inflammation that is going on within the brain completely separate to that, without that peripheral input.
Are they the same disease?
That is the $65,000 question. Probably, because Probably the true MS. Some people, some neurologists have been saying that the true MS is progressive MS. But that in some people, they have a genetic susceptibility that when the progressive MS starts, it triggers the immune response.
I believe I've had MS for 65 years this year, and as far as I know, I have never had a relapse.
Yep, that's...
I mean, is that normal, unusual?
It's not unusual. About 10% of people, I believe, never have a relapse.
Okay. One last question. You're optimistic that you're going to find something that is going to be useful in preventing or remyelinating?
I am optimistic, and I'm optimistic about the field in general. I'm not the only person working on this. There are a lot of people all throughout the world who are trying to develop treatments, and we've been seeing more clinical trials coming up for treatments that are trying to target specifically the local inflammation. And so I'm hopeful that things are going to be going the right way. Mm-hmm.
Brothers, sisters, cousins, and then I know over the years I met one lady, her daughter got MS, and then her granddaughter got MS. So yeah, and there's quite a few cases. People,
yeah.
And I have two cousins that have MS on my mother's side, nothing on my father's side.
So the question...
Might just repeat the question?
Yeah, so the question is about sort of genetic susceptibility to MS and how it can run in families. So we know that there are MS risk genes, and we know from family studies, if you have a twin who has MS, an identical twin, you are very likely to develop MS. I think the number is close to 50%. A non-identical twin, it drops down to somewhere around 25, and then normally for a first degree relative, so normally a parent or a non-twin sibling, the rate of MS is increased only by about 3% from the general population. However, there are circumstances where we do sometimes see these runs in families, and they're being investigated around the world in a lot of studies, and we think that these families are passing on specific high-risk genes to the people, because it's not just a one gene disorder, it's a combination of genes, and so it seems like some families have a particularly high combination of genes, whereas normally it might be just a few.
Michelle's asking, would it be better to advise people not to have children or to put off having children?
The problem is that it's not a simple one gene, one disease, like something like cystic fibrosis or sickle cell anemia. It's caused by a lot of genes, and like I said, most people who have MS and have children, their children's risk is only increased by 3%. I think it's important to inform people about what those risks are, and especially if you seem to have a lot in your family, you may have a higher risk, but I don't know that I would say that people with MS should necessarily not be having children.
How far away, and what are the plans for human trials?
For mine, it's quite a few years away. Unfortunately, we're starting animal experiments this year to see how this is working, and I'm also developing a humanized cell culture system, so I can test all of my things in human cells as well, to make sure that it's translatable. Because often the problem we find is something works in mice, it doesn't work in a human, and so I want to make sure that doesn't happen. I'm hoping 5 to 10 years, optimistically, we'll see.
Are there any other questions? If, oh, yes.
I mean, we all have millions and millions of genes inside. We don't know what genes, and we all may have gene for MS. And then the thing is, what can trigger that start disease? Something must trigger it. That's the question.
Yes, so the question's about what is triggering MS. So it seems that Epstein-Barr is one thing that is triggering MS. We haven't quite worked out exactly what it's doing. It could be that it's just creating the inflammation in the immune system, but it's also possible that some of the proteins on the Epstein-Barr virus look like myelin proteins, and then your immune system gets confused and it attacks the myelin. So depending on how it works, a vaccine for Epstein-Barr might be beneficial for MS. It depends, because if it's that molecular mimicry where the Epstein-Barr protein looks like that myelin protein, then a vaccine might not work because you're making an immune response to those proteins. But if it is more about the way that the Epstein-Barr is like, it promotes B cell, the antibody-producing cell proliferation and stuff. If it comes to that, then a vaccine may actually be able to reduce prevalences of MS. So there are a lot of groups around the world looking at how that is working, and hopefully we'll have an answer on that soon. There are Epstein-Barr vaccines that are in clinical trials, so I'm really interested to see how that all plays out.
A bit louder.
A question about artificial intelligence and how some of these new technologies are being used within MS research to accelerate finding cures, if at all.
So the question's about if AI is being used to accelerate MS research and find cures. And I would say that people are using it to analyze these really large data sets that we produce about genomics, about the genes and how everything is responding. It can be quite difficult to get to the bottom of like these giant spreadsheets, and the AI is being used to sort of pull out patterns that we might not have seen before, and it's also people are looking at using it to find ways to analyze MRI data so that we can maybe find ways where AI can help us track progression better in an individual patient.
It's wonderful to hear that there is real progress, research progress being made, looking at how the cells are regulated and the possibility that we might be able to either prevent some of the myelin damage that's taking place or repair it. And I'll ask you to join with me to thank Sarabeth for her presentation. We do have a small gift for you, which I'll give you in a minute. Thank you.
Sorry, I ran away too quickly.
That's all right. We'll take a brief break while we organise the next speaker. Thank you.